Okay, I think it's on my mobile phone actually. Right now the connection for the laptop is not established, so I am logged with the mobile phone.
No problem, sir.
So yes, please just let me know whenever you would like to start.
Sure.
Or
Sure, sir. Sure. Right now, yesterday it was raining in Freiburg, and the temperature was 25 degrees. But it started raining suddenly in the afternoon, and now it's again sunny.
Okay, so right now what's the time there?
Uh, right now it's 7 o'clock. सर आप तो यहाँ 7 साल थे मुझसे ज्यादा आपको पता होगा. सर हमारे कुछ पार्टिसिपेंट्स जो है वो यूट्यूब लाइव पे है तो yeah, and I think we have got more than 150 registrations for your event, which is very good. So all our participants will be on YouTube live. So and whatever questions they want to ask, they will write down in their chat box. So we will easily get.
Yeah, I think that's more on the precision genomics part that we would be talking about in briefly about my journey. Regarding the immunology part, that's a very small portion of the work that I have done. So as it proceeds during the conversation, you can plan. And I hope most of the people are comfortable in Hindi, I think.
हाँ मतलब आई वुड से कोई भी लैंग्वेज चलेगा ऐसा कुछ भी नहीं है कि इंग्लिश में बात करना है हिंदी भी ठीक है
इवन गुजराती चाल से साहब
अरे so yeah, it's good. We have few people also on Google Meeting, and majority of our audience will be on YouTube Live because all the registered participants would get the link from via email. So and that email has YouTube Live link, which is again unlisted. So it's like a private link. Not everybody can access it. If you receive it, then only you can access that.
It's an interesting series that you have started. Most of the time, people hear about research work and the person behind research is generally not known actually, or we don't know people as person. We see them as some researcher or someone working in the lab, but the journey is also interesting sometimes.
Yes, sir, absolutely, that's true. Okay, so I think we are already live now, and once I get the green signal, we shall start. Yeah, perfect. So we are live now. First of all, I would like to welcome all our audience today who have joined to listen our most awaited guest, Dr. Pushpendra Singh. And I would like to request my teammate Bhakti to please introduce sir for the today's event.
So, hello everyone. I guess I'm audible.
Yes.
Yes.
Hello everyone. So let me quickly introduce our today's speaker, Dr. Pushpendra Singh, one of the luminaries in their field with a profound dedication and relentless knowledge. He completed his master's in biotechnology from Dr. B.R. Ambedkar University, Agra in 2002 and PhD from ICMR Agra in 2008. Then he worked as EPFL from 2008 to 2014 in Lausanne, Switzerland. From 2014 to 16, he was working as a visiting scientist at National Hansen's Disease Program, and then he joined there as a senior postdoctoral researcher. From 2016 to 19, he worked as an assistant professor at the Maharaja Sayajirao University of Baroda, and currently he is working as a scientist at ICMR Jabalpur. He has attended and organized many national and international conferences and is also involved in many professional societies. Dr. Pushpendra Singh is working in the field of microbial genomics and pathogenesis. The research work involves bacterial disease as well as viral diseases. His research interests are microbial genomics and pathogenesis, neglected tropical diseases, viral diseases, research, and diagnostics. He also appreciates the importance of science and communication and actively motivates people to pursue career in science and research. He is talented, dynamic, and diligent person, showing excellence in every sphere of knowledge. With this warm welcome, I now request to proceed further in the event. Thank you.
Thank you so much,
Thank you,
Bhakti,
Bhakti.
for the introduction. Sir, was it correct and was it good?
Mm. Thank you, Bhakti, and there, I think most of these are. I thank you for your kind words, but I think the most important one is that I am very lucky to be in correct place and to get very good mentors throughout my career. I think whatever I can achieve during the journey was because of the excellent mentorship that I got from my mentors from right from my school education till the postdoctoral training. So I think yes, the most important aspect that I consider is that have been very lucky. And rest of the qualities actually are only some reflection of the mentors that I have worked with. So
Yeah.
I'm only a fraction of what my mentors are actually. So thank you for your kind words, but I think that I'm just an ordinary person who was very extraordinary in getting excellent mentorship. Thank you.
That's true. That's true, sir. I think we all need mentorship at a point where we don't know what to do ahead, and then the correct advice. Okay, so let's start the today's podcast, I would say. And yeah, so first thing that I would like to ask, and also many of them would be interested in, is that like how did you came across research? Because not everybody is interested in research, right? So there would have been some spark, right, at some point of your life. Yeah, you decided that I would do a biological research, or I will go into research. So what was that turning point for you? When did you decide that yes?
So thank you. This is a very interesting question actually. I was very much keen to understand biological phenomena. So until 12th, I was still trying to figure out if I should be pursuing mathematics or biology. But then I pursued both of them, and during that time, I knew that my inclination is more towards biology. So after 12th, after going through some of these subjects and very good teachers, I think what we are interested is more of a reflection of how good teachers we get for a particular subject.
Mm-hmm.
And then from a childhood curiosity also, I was more interested towards pursuing a research career. And then I found that biology interests me more. When we study physics, certainly the stars and the distance in light years and Big Bang and everything excites us. But when we dive deep into biology, I think the same excitement also emerges because just four letters can guide so much diversity in the biology in system, or how you basically see so many colors in different life forms, so much adaptation. So understanding all that through molecular biology, or trying to summarize that all this is because of those four letters of DNA or RNA. So that's very exciting actually, and to apply them for disease diagnostics or treatment or developing novel interventions, vaccines,
Yeah.
all this
Yeah.
comes from that basic understanding of biology. So yes, that
Yeah.
I think that, to me this is as exciting as the astronomy to when I was trying to explore excitement of physics. So
Yeah.
biology also has the similar dimensions, but in a reverse manner. You have light years there, but then you have femtoseconds and femtogram amounts and nanometer distances in biology.
That's true. So it is very common to get attracted towards astrophysicist.
Sure, I think from the very beginning, almost everybody is very much inspired because you can see it that stars are so far away when you start
Yeah.
reading. In biology, you have to experience
Yeah.
usually from the textbooks. It's, it's especially microbiology
Yeah.
and
Yeah.
molecular biology. You have
Yeah.
to imagine things and then you have to prove that the imaginations were correct or not, or the mechanisms have
Yeah.
to be established for those.
Yeah. 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 That's true. That's true. Sir, did you ever think when you decided to pursue research that you'll ever become a full-time PI, like an academic
So,
scientist?
mhm. So it's actually through the journey that after pursuing my master's, and that also happened by seeing other master's students that
Mm-hmm.
they
Mm-hmm.
used and at Ambedkar University. So
Mm-hmm.
and during that time, our classes were at the National Jalma Institute for Leprosy in Agra, and those
Uh-huh.
scientists
Uh-huh.
were so dedicated that they wouldn't compromise their own research work, but they would also want to contribute to the academics and to the MSc program that was started
Mm-hmm.
at that
Mm-hmm.
time. So they
Mm-hmm.
used to take our
Mm-hmm.
classes at 7 in the morning. Uh, our university was on the one side of the city and Jalma was on the other end of the city. So we had to cross uh, to reach there at 7 a.m. Till 7 to 9 a.m. we would be taught by the directors and deputy directors and other scientists of ICMR.
Mm-hmm.
And that was actually very much interesting that how keen they were, that they were very much committed towards the academic growth of the students from Agra University.
Mm-hmm. Mm-hmm. Mm-hmm.
This is amazing because when we start complaining that our timings are not suitable or we are not getting enough attention, but all those people actually found time for us to check our copies, to set papers and everything from their full-time research schedule.
Yeah.
And that helped us a lot actually. I would say that
Mm-hmm. Mm-hmm.
that shaped career of so many students, and we sometimes fail to recognize the sacrifice of our teacher, how they pursued their research career as well as teaching. In university
Mm-hmm. Mm-hmm.
setting, I found it very hard to balance research and teaching. That was
Mm-hmm. Yeah.
perhaps one of the reasons that I chose to come to ICMR for full-time
Yeah.
research. But then you imagine that how our MSc teachers in Jalma, those were also
Uh-huh.
full-time scientists, and they were
Yeah.
managing it uh, so well, and we are really grateful to them.
Yes, sir. That's very much important. I still remember we were studying in CMB department, and we also had to like take lectures from other faculties, and one of them were you as well. And then we contacted you that if you can please teach us this subject, because you were teaching since long years in the university.
Thank you. I think this is because we were taught by our teachers from their extra time. So
Yeah,
when we start comparing or binding ourselves into certain timings, then
yeah.
it becomes more of a, yes, certainly more professional life.
Mhm,
But
mhm.
when we want to contribute to society and give back, I think we have to find times and ways for that. And
Yeah, that's
that's
true.
very satisfying when we see people uh, growing. And if we have made a slightest contribution to their growth, that's very satisfying.
Yeah, that's true. That's true. Okay. So now moving a little bit ahead into your journey, I would like to ask a very, I would say, very simple thing, that why did you choose leprosy as part of your PhD later on? Like what sparked your interest in specifically in leprosy, that you continued your journey for PhD as well after your masters?
So my master's dissertation was on mycobacterial drug resistance mechanisms, particularly on
Mm-hmm,
streptomycin resistance.
mm-hmm, mm-hmm,
So as I said that we used to get limited time only in the morning, 7 to 9 for teaching or for our classes, and some
mm-hmm,
practicals were also only on the weekend. So
mm-hmm,
some of our content couldn't be covered because for example, bioinformatics was very new
mm-hmm,
for us. And during
mm-hmm,
the dissertation, I saw one research paper related to my dissertation work from
mm-hmm.
Professor Stuart Cole that was on streptomycin drug resistance. I still remember that was a paper in antimicrobial agents chemotherapy in 1994, and that mentioned the gene of this RPFL gene that is the target of streptomycin.
Mhm,
So I started looking at that gene manually, and then the data that I got for the sequencing from our the dissertation work that I carried out, and those sequences were matching 100% exactly one by one, A, T, G, and like that. So
Oi.
now we have so many softwares to align them automatically and then correct the sequences through chromatogram. But
Mhm.
I was doing it manually
Oh
because
my God.
those tools were not available, and I was fortunate that it was the entire gene available to me, so I could see it. So that actually made me interested in microbacterial drug resistance mechanisms. And
Mhm,
my
mhm.
PhD work was on tuberculosis drug resistance. And during some
Ah.
of those meetings which were conducted at our institute, one was informal consultation of WHO on rifampicin resistance in leprosy. Uh,
Mhm, mhm,
So at the time, I was mainly working on tuberculosis drug resistance, but since this was an event where Professor Stuart Cole himself was coming, and he had been the pioneer in genomics, especially of mycobacterial diseases. So I got a chance to attend his presentation, and I asked some question about what is the fitness cost of rifampicin resistance. So he gave certain he explained that, and when I was almost finishing my PhD, I contacted him. I continued my interaction with him by sending my research publications, and
mhm,
it
mhm,
so happened that he was also planning to move from Institute Pasteur uh, to Switzerland at that time to create a new research group, and there interact. I sent my application to him, and I was fortunate
mhm,
to be in his group. And at that time, since my initial interaction with Professor Stuart Cole was about leprosy drug resistance, so he gave me a topic
mhm.
on leprosy genotyping and drug resistance mechanisms.
I
He
see.
had a similar project at that time, so that's how basically I became interested in microbial disease, and then it evolved into drug resistance in leprosy and genotyping. and that topic has actually been very interesting. So I have pursued it that since then.
Great, great. Sir, so you were talking about I'll go step by step. There's a lot of information that is very interesting. So I wanted to ask that you said that like during your masters you had to do gene sequencing by yourself. So was it due to the lack of softwares? Like you didn't have any softwares, or people used to do like this, or you were more interested in learning it manually?
Mm. No, it's actually that it was in 2001 and 2002.
Mm-hmm.
So to have access to internet and to all those softwares and resources, even the soft copy of our sequences was difficult to obtain.
Oh
So
my
we
God.
got a printout of the chromatogram to attach in
Oh.
our dissertation thesis, and that was the only source that I can check. So that is why I basically found I had to find ways to analyze the data. There are ways of doing it automatically, but as I mentioned that we used to have limited understanding or teaching of bioinformatics at that time. So I did that
Yes,
manually,
that's
but
true.
that went well actually. One mutation that I found was present in reference strain itself, and I informed this to my supervisor, Dr. V.M. Katoch, and we were not sure how the reference strain can have that mutation. But when I joined Professor Stuart Cole's lab some years later, uh, and I discussed this with him, and it was identified that there were some 67 errors in the reference strain also, and we were actually seeing at that sequencing error in our data from the reference strain itself. So sometimes we have to believe our observations also. It's not that uh, things cannot be updated. We have to see that whatever we are observing is also correct or not.
Yeah, that's true. That's true. Sir, so if the gene is of, let's say, like, for example, one kb or 1,000 base pairs, then you used to literally like align all the 1,000 base pairs.
So it is that was only a 375 base pair gene.
Ah,
So
okay.
yes, I aligned it one by one. I aligned
Okay.
it one by one, and that was, I think, the most exciting experiment or the bioinformatic analysis I carried out. It's certainly not scalable. If you have only 7 or 8 results, that is okay. But now we
Yeah,
can be more efficient with the availability of tools. But
yeah,
what surprises me is that even today, when we have access to all these softwares and such things, people are not very willing to complete their analysis in time-bound manner to publish it.
yeah,
So
yeah. That's
it's
true.
been 22 years since then, and if we see that there have been several generations that have changed or now we have to
Yeah,
update. So accordingly, software availability and everything has been internet and everything has been much more easier now. So we have to be more efficient in analyzing our data and publishing those timely.
that's true. That's true. And sir, you also say about your PhD that you did it on tuberculosis, right? So
Yes.
so so during that time you were also connected to Jalma Institute, right?
Yes.
So during that time, like what was the, I would say, the biggest hurdle during your PhD? Like, do you remember any incident that you might have ever thought that like this was like one of the thing that you surpassed by yourself and you could achieve it?
Mm. So we were the first batch of students to join PhD uh,
Uh-huh.
from our university. There were other permanent staff who were carrying out PhD before us. So for them, things were different because they were permanent staff, so they could devote more time for their PhD. But those of us who had joined with our fellowship, we had to actually finish things in timely within our fellowship period itself. So having a batch, like we were 6 students who joined in same time for PhD. So helping each other was very good actually. We were able to help each other when there were some difficulties. There were other challenges at that time, or those appeared as challenge at that time, that we have to contribute to several other things in the department, for example, diagnostics, or providing resources to other laboratories from our micromaterial repository. We used to sometimes think that it's a lot of work, which may not appear directly related to our PhD, but those connections through those type of supplying reagents or helping patients or clinicians in performing diagnostics and drug resistance testing, all those were very helpful actually in subsequent career, because we were able to learn the importance of clinical sample processing, identifying what can be the errors, what are the best ways of finding correct sensitivity profiles, molecular mechanisms, and especially multitasking. So all those things at that time appeared very stressful, but I think that was a training, and all PhD programs are actually training. You devote your maybe 20% to 50% time on your PhD topic, but remaining time you are basically learning how to collaborate, how to help others, how to get help, or how to learn things which are not available to you. Or you can get help from nearby lab or from nearby institution, or even from someone who is collaborating with your PI or supervisor. So all those things were very helpful for us in our subsequent career, about writing proposals, compiling reports, organizing conferences, or receiving guests from railway station or airports. All those things actually are opportunities to interact with some of the eminent scientists, and some of those interactions can actually change your career in a very different direction, depending on what type of interaction and whether you are present in right time, in right place. So I think all those challenges at that time used to look very difficult, but when you look back, all those difficult times were not actually so difficult actually. When you look back, we sometimes react to some things in a very intense way, and those really appear intense in that moment. But I think things go well if we have perseverance to complete those. And for all PhD students, I would say that yes, it's a training actually. We have to learn to overcome some of the difficulties. If there are no difficulties, that means the training is not good enough.
That's true. So I really take these words that like focusing on a topic is like, okay, 20-30%. Rest of the time you are doing other things which are also important. Sometimes we feel, me being a PhD student, I also feel like doing other things are like no, not much good. But I think I have an alarm. Sorry.
So I would just continue to say that this 20 to 50% time for your PhD, that may depend on different situations actually. I was telling about our first batch who had to also prepare the ecosystem for PhD because administration was not geared according to our needs. Because for them, for the admin also, even though they were supportive, they were not geared because it was also first experience for them.
Yeah.
But with the help from our supervisors and teachers, so things went well at the end. Most of our colleagues were very well placed actually. Even though at that time it looked like we are not able to continue very good research, or at that time
Yeah.
it looked like we are doing routine stuff for diagnostics and such things. All those things actually matter because those are assets, the resources that you generate, the interactions that you build, all those turn out to be resource and asset later on.
That's true. That's certainly true. So, sir, since you
In some
mentioned
of the
about,
laboratories, yeah, I would say that in some of the laboratories you are expected to spend more time on your PhD work. That also
yeah,
depends on the lab culture and the lab infrastructure. So yes, if you may be expected to devote more time, like even
yeah,
more than 60-70% of your time on PhD if your lab is
hmm,
uh,
hmm,
giving
yeah,
so much priority on that particular topic. But at that
hmm,
time, you have to also learn other things.
that's true. That's true. So learning soft skills are also important because, like, later on, if we finish PhD, we're gonna interact with others in the community, and if we do not make our connections, that then would be a problem for us only.
True. Yes.
Sir, so since you mentioned about your PhD and you were the first batch, like, what was that one thing that you still remember from your PhD? Like something like one of the best experiment that, okay, because I did this experiment, I got this publication, and you were like, I'm very happy out of this. So what was that one like pivotal moment that you would remember till end of your life?
So, as I said, we got the one instrument called MGA. That there was an automated liquid culture system that we
Uh-huh.
got, Bacti Alert, and a similar publication about comparing the sensitivity profile using this automated system and the uh, other manual system was published in a journal which had at that time impact of 0.5 or something less than that actually.
Mm-hmm.
So I
Mm-hmm.
designed my work using taking that paper as a model paper and
Mm-hmm.
performed the experiments, and that was on paragenamide sensitivity testing. Paragenamide uh, sensitivity testing is considered difficult because it is pH dependent, and at that time um, when I completed that work, my PhD supervisor suggested to submit it to Journal of Clinical Microbiology, and I was not sure that it would get accepted there or not, but
Mm-hmm.
that was the first publication from our our institute in Journal of Clinical Microbiology, and that that was very that was actually very good to see this publication just in time when I was about to complete my PhD work, and that paper helped me in getting some communication started with Professor Stuart Cole that led to another opportunity. So I
Mm-hmm.
think believing that we can start with higher targets and believing and having some supervisors or mentors who can support you in
Yeah.
seeing higher goals. That is very good actually in the early stage of your career. Similarly, uh, with this opportunity, I got a chance to work with Professor Stuart Cole, and at that time when we were working on armadillo
Mm-hmm.
having natural leprosy, so whether this is
Mm-hmm.
zoonosis or not, that
Mm-hmm.
we were that we were trying to investigate, and it was
Mm-hmm.
very well known from last 20 years when we started this work that armadillos carry natural leprosy infection.
Yeah.
But our evidence was uh, based on the whole genome sequence, and that was accepted in New England Journal of Medicine,
Oh.
which is a reputed medical, journal in medical.
Yes.
So at that time also our Professor Stuart Cole, he said that let's try in that journal, and earlier reports were published in very small journals, but this journal has a very high impact in clinicians and medical practitioners.
Yes.
So
Yes.
having that journal published in, having that study published in New England Journal of Medicine was covered by media worldwide,
Yes.
including newspapers in different languages. I saw it in Rajasthan Patrika also after just the embargo
Oh.
period was over. In Bhaskar and in Hindi newspaper, they had mentioned armadillo as a cheaty core. So because they translated it as per whatever people would be able to understand, because armadillo is
Yeah.
not a common name in India.
Yeah.
So
Yeah. That's true.
seeing that our work reaching in form of a newspaper at my home through this study was truly inspiring. And then seeing the coverage in Facebook, LinkedIn, where people were changing their behavior for not eating armadillos, because at that time, until that time, it was not established whether this can cause leprosy to humans.
Yeah. Yeah.
So
Yeah.
when people change their behavior, that is the real impact of any research.
Yes.
Or when people notice that finding and accordingly start taking certain behavioral changes, that's really good impact of science.
Definitely.
Those were
Definitely.
memorable.
Definitely, sir. Sir, how did you feel when your paper got published, like in New England Journal? Like, what was your first response?
So when these findings were published, I was my Professor Stuart Cole informed me through email, and I was in India at that time. So I think there was some minor comments that were to be addressed, and it was already clear that this is going to be published. Suggested just some minor change in the title about using probable genotic leprosy rather than zoonotic leprosy. So
Yeah.
I think it was also the time when I was in India for my marriage actually. So together it was very memorable that I got that news at that time.
All the stars aligned together.
Yeah, I think those things actually help us get good collaboration. Later on, we published ancient genome study with another group in Germany, Professor Johannes Kraus group, and that ancient genome sequencing of leprosy through those 1,000-year skeletons was published in Science 2 years later. So that was also wonderful to have this type of collaboration, because one success opens ways for other successes. So this
That's
is,
true.
and certainly there are so many failures also, but the good thing about failures is that not all failures would lead to subsequent failures. If you are
Yeah.
taking lessons from your failure.
Yeah, that's true.
And then once you get onto the path of success, then those success further help or make it easy for getting another success.
Absolutely, sir. Absolutely. Sir, so since you mentioned about, having mentors in life, you said that you had a lot of mentors. So would you like to say like that at which time of your life you required like mentor at I mean I understand you require in the end of your journey, maybe even now. So like how do you choose correct mentor for yourself? That would be my, I think, appropriate question.
I think just like we do not choose our parents or family,
Yeah.
we always do not have choice of choosing mentors actually.
Uh-huh.
But whoever are available to us, uh, we have to find which one are most optimal as per
Yeah.
our career aspirations and whom we idealize. Uh,
Yeah. That's
where
true.
if somebody's hard work is dispelling us to not pursue that career or that lifestyle, then certainly we would not be willing to take them as uh, mentor.
Yeah.
But we have to see uh, what we want to achieve from our career, and
Yeah.
in that direction, who can be our ideal mentor.
Yeah.
For certain aspects such as PhD mentors and those, we, we, we are not always able to say that. But for even with that, we can have at least two or three mentors actually. For
Mm-hmm.
we can have someone who can give us guidance about our social life or for our um, other extracurricular activities, or
Mm-hmm.
whenever there are some personal problems,
Yeah.
we should be able to discuss with uh, some people. Those may be our friends also. Uh,
Mm-hmm. Yeah.
I think to gain someone else's perspective is very important.
Yeah.
Something that may appear so much overwhelming to us may be a very routine procedure or routine situation, or
Right.
someone else might have seen it uh, before us and might have handled it very well, and they may be giving us some clues uh, which may be relevant for our situation. So all those things we have to uh, we have to be very open for others' perspective, and I think there is no alternate of sincerity and hard work, especially
Yeah.
in career in research. It's competitive. What I heard from
Yeah.
my mentors and other successful researchers, I
Mm-hmm.
believe that it's very competitive. Being good is not good enough in if you want to excel in research.
Yeah.
You have to put something extra than the ordinary to to gain the extraordinary result.
Yeah. That's true. That's true. So, do you still have some mentors in your life
Yes,
at this
I
time?
think I'm very lucky to have very good colleagues, but at some points we need mentors also. And
Mm-hmm.
recently I approached few people uh, out of my immediate work also for mentoring me. Some of those were my uh, teachers or researchers in previous workplaces.
Mm-hmm.
Some
Mm-hmm.
I have interacted during the COVID time in the medical
Mm-hmm. Yeah.
college, and there are some others who are just by casual interaction they are mentoring me for uh, time management and
Yeah.
uh, overall well-being. So I think we
Yeah.
have different aspects of our life where we can get some help from others.
Yeah. That's true. That's true. That's true. So now moving a little bit ahead in your journey. So like, how was the transition from India to Switzerland, and like how did you adapt to it, and like how did you started your research work? And I would like to know the story, that how exactly the time flew with you when you decided to do a postdoc after your PhD.
Mm. So when I was about to submit my PhD. I had approached Professor Stuart Cole, and just 2 days after my PhD viva,
Hmm.
I I had to join Switzerland, and so that was certainly most memorable experience of my life, and most English is also not among the first 4 official languages that Switzerland has. So to adapt to those, that atmosphere, but it all went so well and so much memorable. I think people are very cooperative. So that also made it very easy. People were very open, and I also learned that you can be very happy and very productive,
Mhm.
and very hard and remain happy and be
Yeah.
productive. There is no contradiction in that actually. But also when there are deadlines, we have to meet those deadlines and we have to give our best rather
Yeah.
than restricting to ourselves into certain boundaries. And when we
Yeah.
have that leaves a lot of scope for celebration and enjoying our success also.
Yeah.
Yes, that was wonderful to have that opportunity, especially that Professor Stuart Cole, Dr. Richard Truman, whom I have worked with, and then my PhD supervisor, Dr. V.M. Katoch. All those people were very hardworking, but also very ground to our, very much connected to people, very, I think, very friendly. Sometimes some people may express it, some may not, but those were always very approachable and very helpful and very concerned about our career growth. Sometimes it may
Yeah.
be that some people are very strict and they may be uh, they may appear harsh, but we have to see that what is the real intention of that person. And if we trust that person, we should not mind few such things that we were scolded or what was told to us in front
Yeah.
of others. Because sometimes it could be that particular moment, but it's more the longer consequences that we have to see. Consequences or whatever is the long-term benefit for us from those
Yeah.
interactions or opportunities.
Yeah. That's true. That's true. Certainly. So, also any any memorable again the moment in during your postdoc, like what did you feel when you came from India and then you joined the lab in Switzerland? What difference did you see at the first place? I mean, I understand maybe better facilities and better instruments and all, but apart from that, how did you feel?
Mhm. Uh, I thank you for taking me back in time. I think people were very open to help and prepared actually. If,
Hmm.
uh, for example, if you have to get some cards for your residency there, those
Yeah.
instructions used to take only few minutes. Uh,
Yes.
there were because they had everything prepared or in basically everything was kept arranged in a way that it will not waste so much time in. For example, for ordering of reagents, you just have to write somewhere in the book, and
Yeah.
someone will order it and place it on your desk in
Yes.
just a week or two. So all those things were unimaginable to me at that time, uh, and that leaves room for doing research uh, more seriously or more to spend more time on your research work. Uh,
Yeah.
so that was one of the most memorable initial experience. People were also very friendly, but then I also realized that when we uh, we have certain ways of giving and taking rights on each other. So we are
Mm-hmm.
very
Mm-hmm.
insistent on certain things about when we consider someone as friend. But I think that may be a different culture in different places. So we have to
Yeah.
adapt to that culture also, and it took some time for me to figure out that I cannot be very insistent on inviting somebody.
Yes.
Yeah, available.
Yeah.
So all such things in India, we are very much insistent. कि कैसे नहीं आएगा तू? So all those things doesn't work in every culture probably,
That's true.
or it may take time actually.
Yes.
Yes,
Absolutely.
those type of things or those type of bonding take some time.
Yeah. Yeah. Absolutely. And also taking care of the allergies that we need to take care from our research group, that who has which allergy, because our Indian food has all the
हाँ,
spices.
किसी को कैट से एलर्जी है, किसी को डॉग से एलर्जी है, तो ये सब बातें फूड एलर्जीज और ये सारी चीजें भी
हाँ, मतलब मैंने ऐसे लैक्टोज इंटॉलरेंस सुना था जब मैं इंडिया में था, बट यहाँ आके पता चल रहा है कि ये सच में एग्जिस्ट, मतलब पता था कि एग्जिस्ट करता है, लेकिन यहाँ पे लोग इतने ज्यादा कॉन्शियस है कि दूध भी अलग होता है.
हाँ, वो सब चीजें.
Exactly, sir. So, like, would you like to, since you mentioned about your paper getting accepted, and like, of course, it was everywhere on the news and media as well. So, would you like to talk a little bit about the scientific part of that paper and what exactly did you do, and what was your contribution in that?
So, firstly, about this New England Journal of Medicine, when this paper was published, the evidence was already there that leprosy infection is present naturally in armadillos. But whether the strains are same or not, that was not established because leprosy genome is so conserved, so similar between different strains that until you do whole genome sequencing, it would be difficult to prove that they are just same genotype but really the same strain or not.
Yeah, so, that's true.
Develop ways for whole genome sequencing. I think so. I not that help the this was interesting actually to complete it, and I think that helped in its acceptance.
सर सॉरी, आपका आवाज पूरा कट गया था.
हाँ, ओके. तो मैं यही बोल रहा था कि जब हमारा एविडेंस इतना स्ट्रॉन्ग था कि दो स्कीम्स को हमने जब VNTR और स्निप जीनोटाइपिंग को मर्ज किया, तो अब उसको किस तरह से बेसिकली स्टोरी को लिखते कैसे हैं वो काफी इम्पोर्टेंट होता है. So how well you write that. So I think that if similar work can get published in a very reputed journal also, if you are interpreting the results and writing and conveying them to the readers. So yes, that was good.
I see. Okay. So I would definitely consider this being the one of the very best highlight of your research journey. Yes or no?
Surely it was, but subsequently the publication in Science was also very helpful, and it also had its own challenges. So I think collaborations are very important from different disciplines. When people from archaeology, bioinformatics, leprosy biology, and those who are working on radiocarbon dating and mycolic acid profile. So when they come together, then they interpret results and findings from different angles and produce evidence which cannot be easily challenged. For example, contamination is a very common criticism about any ancient DNA research. But when you have evidence regarding characteristic mycolic acid profile of M. lepri from those skeleton samples from which are 1,000 year old, then that criticism of contamination cannot stand true. So then the same criticism that it can be a contamination, that you can prove based on the DNA damage patterns of ancient DNA. So you have to sequence twice ancient DNA without repair of the basis and ancient DNA after the repair of the damage pattern. So then you compare that this many percentage of bases were damaged, which can only happen in the ancient DNA. It cannot happen for the modern DNA. So all those difficulties actually can serve as challenge, as opportunities to validate your findings, and that has helped the field of molecular biology, I think.
Absolutely.
Last year's Nobel Prize to Dr. Swante Pabo was on the ancient DNA research of Neanderthals, and Dr. Johannes Strauss was his postdoc, who actually was joint corresponding author in this study of ancient DNA in leprosy also. So all those challenges that came during the way were addressed by developing, and that helped us in publishing in science. Then later on in the proceedings of National Academy of Science, PNAS journal of USA for the lepromatosis genome. So those few publications were one after another. Every 2 years we were able to get these findings published, and it was because of collaboration with several of my colleagues, friends, and of course mentors who guided us and helped us in writing those so well that they can get published.
Yeah, of course. So, I would like to ask you like one question that. So I have three sets of questions for you. So first set of question was that what what advice being a PI now would you like to give to the young postgraduates who are just finishing their masters or they are pursuing their masters?
I think that one should be willing to work hard and to enjoy the work that they want to pursue. If somebody, so that it's very important to be happy while doing what you do. Otherwise, if you consider this as a job or as a work or as a difficulty, then you would keep on working as a worker. But if you want to really be a researcher, you have to be interested in the topic or in the subject, and you have to basically enjoy your work. Otherwise, there are so many other things where you can get paid better. But if you want to be a researcher, you should really be keen to explore new things, to understand what has not been understood by anyone else before, or to generate new data. I think working hard is important, but staying happy while working hard is even more important.
That's certainly the advice that I would really consider in entire of my career. And also, what would be the advice that you would be giving to the postdoctoral researchers? Like maybe I would be doing in future. I don't know. Hopefully.
So I think the postdoc time is really the best time of our career. if you see that as a PhD student, you have certain limitations. You have a very very important priority of completing your PhD because it's a
Yeah.
degree and it's also time bound. But
Yeah.
postdoctoral work, you have a PI who is sparing time for arranging reagents, who is arranging your salary, who is actually arranging instruments, all the
Yeah.
logistics and such administrative things, renewal, advertisement, who is also providing you technicians and all necessary infrastructure and ecosystem. You just have to utilize that to the best of your ability, and then also at that time learn how to run your own lab when you would be PI yourself.
Mm-hmm. Mm-hmm.
So the most active research and most serious output can be actually expected during their postdoctoral journey.
Mm-hmm.
Uh,
Mm-hmm.
and at that time, you can also figure out whether you really want to be a PI or not, because
I see.
you get to see the challenges, you get to see
Yeah.
whether this actually is field for you or not,
Yeah.
and the sooner you figure it out, you can accordingly plan to be a PI or not, and
Mm-hmm. Mm-hmm. I see. I see. So since you mentioned this, really a crucial thing that like once you are in a postdoc now, then you get a chance to decide whether you want to become a academic PI or academic scientist or not. So how was your transition during that? Like because you were also getting married at the same time, you were maybe somewhere in the postdoc in some country, and then what did you think? Did you come back, and how was your transition so during that time?
so I would say that from the postdoc shouldn't be forever, and that is the reason that many institutions or universities have certain rules that you can stay postdoc for 5 years in this particular institution, because
Mm-hmm.
you have to consider other factors about the age limit in several places.
Mm-hmm. Mm-hmm.
You have to also see that uh, you have gained enough experience of writing proposal and
Mm-hmm.
running your lab, uh, or evaluating manuscripts, writing manuscripts.
Mm-hmm.
So after that period, for my case, actually, I was sure that I want to come back to India. Uh, so at that time, I started applying for various fellowships in and positions in India.
Mm-hmm.
And during that time, I got an opportunity to work in the US also. From Switzerland, I went to US for 2 years, and
Uh-huh. Yeah, yeah.
uh, some research question that came in my mind during my uh, work on armadillos, natural susceptibility to leprosy, and then
Mm-hmm.
there was some report saying that some armadillos do not get leprosy even after
Mm-hmm.
experimental infection. So what
Mm-hmm.
makes them resistant was the question that I used to wonder
Mm-hmm.
about, and we developed one research proposal uh, to work on this project in USA,
Mm-hmm.
and I expanded that for my Ramalinga fellowship also, and that
Mm-hmm.
helped me gain that fellowship to come back to India. And
Mm-hmm.
later on, that also gave us some extramural fundings from Netherlands, Leprosy Research Initiative, Netherlands,
Mm-hmm. Mm-hmm.
and that helped me my transition from Switzerland to USA and then to India. Uh,
Mm-hmm. Mm-hmm.
I never chose things based on proximity to my hometown or to the convenience of city or place.
Mm-hmm.
I was very much aware about the reputation of MSU. So instead of planning to come to a nearby city in Rajasthan, which is
Mm-hmm.
closer to my hometown, I chose to come to MSU Baroda. Uh,
Mm-hmm.
and so we have to sometimes see that what aligns well with our overall long-term goals rather than immediate
Mm-hmm.
convenience.
Mm-hmm. Sir, so how was your experience in MSU Baroda?
I think I am still very much amazed with that experience. I really enjoyed it. Uh, I saw that so many batches passed in front of us from different subjects, and I actually wanted to do my MSc from MSU Baroda while I was teaching there. So, but there was probably no such option. So I was sure that a lot of which are being covered in MSU were not available to us when I was doing my MSc. The teachers are very well trained from reputed institutions. Infrastructure has been much better at MSU than a lot of other universities. But also at the time, I used to see some challenges for students. For example, resources. We used to give them tips by counting that you can get 100 tips for your experiment. Because the resources are limited, but then we also, that makes us aware that we have to value resources. So those challenges may appear difficult at that time, but those also give us good lessons actually. Later on, when we are not actually limited by the number of tips or reagents, we have to also value that everything comes at some cost. Somebody arranges for those. So we shouldn't be wasting our resources. And those learnings actually come from places where resources are scarce for certain things.
I see.
And I really enjoyed academic atmosphere there. We used to have these journal clubs and presentations twice a week, and those type of things I miss here. Now we have one MSc course started on my medical health, on medical entomology, masters in medical and public health entomology here. So I get chance to teach here also. So overall, I think that experience would be very helpful in contributing to teaching responsibilities at ICMR also.
Yeah. That's true. That's great. Sir, so like how do you envision your research in like next 10 years, let's say? Like, do you still want to publish in science? Of course. But like, how do you envision it? Or
Yeah, I recently visited IIT Indore, where one of my friend, Dr. Hemchandra Jha, is associate professor currently, and he introduced to several of his colleagues about those papers, and it's been long that I couldn't think of the work that I carried out in my postdoctoral work, because here the immediate priorities were to provide diagnostics for different diseases, or to arrange for the day-to-day lab work, or for the external quality assurances or WHO accreditation process and such things on a daily basis. So surely my main goal to come back to India was to be able to do this type of research in India. We have optimized some of the assays. Certain challenges were there initially, and I think it's very good to have a culture in the laboratory where people are committed to a few topics of their research, and that basically mainly comes from having good PhD students actually. So
that's true.
And we have to make PhD students' life easier also, and that's the responsibility of PI. Some of the things are beyond our control, but in those times we have to arrange that things do not stop for them regarding procurement or such things. Once those things are set up and they are regarding their PhD registration, so those are certain difficulties so far. But now with the ACSIR collaboration, we hope to have more number of PhD students joining our institution, and that will help create that ecosystem of serious research on understanding basic aspects of microbial pathogenesis, genomics, and developing tools for analysis of the data in the real time.
That's true.
Yes, surely we hope to improve our publication quality in coming time.
Sir, I wanted to join PhD and I wanted to join PhD under you. And I think I was still in contact with you, but I got an opportunity here in Germany before I approach you. So I was like, okay, I wait.
That's good that you got this.
Sorry?
That's good that you got a very good opportunity in Germany. I think people should certainly explore opportunities during their MSc time itself, because surely you get a very good exposure abroad also.
Yes.
But I have seen a lot of people waiting for far too long to get admission in PhD abroad. And my simple advice to them is that we need not to wait or spend time in waiting in India. We should register ourselves within maximum 1 year of our completion of our MSc, because there are always opportunities abroad for postdoc also, which is even more flexible actually for several laboratories to consider as a postdoc. So, a lot of our MSc students are still applying abroad, and it's been 5 years since they did their masters. For such incidences, I feel that they could have completed their PhD by now and would have been much better to avail postdoc abroad. Surely people should explore opportunities abroad also, but not at the cost of wasting 3-4 years of their career.
That's true. I absolutely agree on this. I was quite lucky as well. I would say that everything aligned, that like before I finished my master's, I had an offer letter. So pretty well it aligned with everything. So a bit lucky. Yeah.
Any other things that
I think there were. I think I would like to ask one. I think I would like to ask one last question, and then there are like 1 or 2 questions from audience. Like I would choose few questions. So the one last question that I want to ask is that what is the best discovery of science for you?
best discovery in science
Yes.
related to my research work?
Anything is fine. Like you can say random thing as well.
If I basically regarding the overall science, they have different opinions, but fascinates me was the one that I mentioned, that so much diversity through evolution, random evolution. Yes, it took billions, actually the billions of years to achieve that diversity, but understanding that we are so common at the very basic level, we see that a plant may appear so different to us, or an insect may appear so different to us, but they also operate similar mechanisms when it comes to the cellular level. So
that's
unity, that unity is wonderful actually, and that happened by chance is so amazing to because so that is the best explanation that we have, that everything happened through random events, and then the selection of the best fit. So I think if things can emerge from almost nothing to to this level of diversity just by time and chance. So some of the problems that we oversee, we should actually uh, believe that yes, things will evolve. Yes, it will take some time, but yeah, so that's the most fascinating thing that I find about biology. And regarding my research, I think that applying some of this basic research for diagnostics or designing some tools which can make diagnosis easier or can help in developing new drug targets. That's wonderful actually. For example, when you compare different genomes, it may be fascinating for you. You can calculate what is the million years difference between two bacteria. But can you identify certain region which is present in multiple copies in that genome, which will make it easier for the diagnosis? Or maybe can you diagnose four different viruses or bacteria in the same experiment by developing probes which are unique. And the same thing has been done for the COVID diagnostics in real time that they develop they develop so many assays. So I think
that's true.
very fascinating.
Yes, that's true. Sir, so we have one question, a little bit scientific one, and I would also like to ask that. So somebody is asking about that how to diagnose the novel biomarkers in asymptomatic leprosy disease.
Thank you for this question. Actually, leprosy is with a very long incubation period, and it's difficult to say that to define asymptomatic leprosy is difficult. We can say that preclinical leprosy. There's somebody who is about to develop disease, but there are no apparent symptoms. So let's say subclinical or preclinical, because and through some studies, what they have found that, and in one of our research paper also on armadillos infected with after an experimental infection, we found that certain genes start differential expression, and those are related to protection against mycobacteria and cholesterol metabolism is one pathway that was identified to be differentially effective.
I see.
So
I see.
Similar studies were later on confirmed in patient samples also by another group by using comparative transcriptomic approach using RNA sequencing. So those are the markers that have been so far shortlisted, and in few studies from Dr. Anemic Gallu's group, they had described that you can detect certain leprosy reactions 5 years, even 5 years earlier than their onset, because they had collected samples from different time points from the household context of patients. So by comparing those longitudinal samples, they were able to detect certain biomarkers in patient samples 5 years ahead of the actual presentation of the disease pathology of leprosy reactions. So yes, through RNA sequencing or the comparative transcriptomics, one can identify biomarkers which are able to predict the development of disease. But then there are so many host factors also involved, which may translate to other ethnicities or not, that need to be evaluated.
I see. Sir, so there is one more question. So somebody was asking like the differences in the postdoc, but I would like to slightly modify this question and ask you that, like, what difference do you find for a postdoc in India, in Europe, and in US? Because you have experienced all the three academic places, so I think this would be very good to ask. And what is your takeaway?
About general feeling is that one should, if they are serious about pursuing career in research, it's good to have a postdoctoral tenure, either in India or abroad. And in all places, the basic thing is that you have to get a training to be whether you want to be a PI, and if you want to be a PI, then you have to contribute in overall running of the laboratory, so that lot of things when you have to do yourself, you would be better prepared. In regarding different countries, I would say it all depends on what type of PI and the research group that you are in. Some people, so it cannot be actually generalized based on the countries. If I have to simply say that maybe in some countries such as Europe, or especially in Switzerland, you would have more resources available. But then to make the best use of the resources is what counts actually. Superficially, it's so different in India, US, and Europe. But as I mentioned, on a cellular level of that, there is so much similarity again.
Yes.
So we have to actually see it case to case basis. Some laboratories might be very productive and still very happy. Some might be productive but at a cost of happiness, and some might be not happy and not productive also, and that happens in all countries.
Yeah, that's true. Certainly true. So I think one of our member, our admin member, Surajit Pal, he is doing his PhD in Kiel at Max Planck Institute of Evolutionary Biology and University of Kiel. So I think he had some question for you for antimicrobial resistance. So I think I will ask Surajit to please go ahead.
Ah, thank you so much, Dhaya. Thanks for giving me the opportunity. Hi, hello, sir. So basically, since you have been working in the direction of leprosy, at the same time, you also worked in the AMR evolution. For example, if I'm saying about the paper that you published in the with Klebsiella beta-lactamase genes that you have actually looked for some of the isolates in India. So have you actually observed any of the patterns in AMR evolution versus the virulency? Whether an isolate, if it's getting more resistant, is it doing better in virulency? Have you seen any pattern?
So regarding this question, so Klebsiella work was done at MSU, mainly led by Dr. Devashri Gajjar. It's been observed that when there are certain antimicrobial resistance through plasmids, then there is a possibility of acquiring other genes also, which might affect the virulence. So it depends on the bacteria and the plasmid that has been acquired. But in case of mycobacteria, where most of my research work has focused, the role of plasmid has not been actually considered so much important apart from the ulcerans, where there are certain plasmids affecting the virulence. But regarding the evolution of AMR, especially about several pathogens, actually there is a cost of resistance. For example, rifampicin resistance in lot of bacteria has certain survival fitness cost, and bacteria has to evolve certain compensatory mutations to relieve those fitness costs. Usually, the resistant bacteria are slower in their replication.
True.
Until they
have a very slow growth rate.
Until they are able to relax it through certain compensatory mutations.
True.
So yes, it depends on what type of gene target that has been mutated for resistance, whether it is a secretory molecule, then it can also affect the virulence also.
So basically what you are saying, it depends on the kind of mutation that it has occurred in the course of evolution, which may
Yes.
lead to the virulence property.
Yes. Certain
Is
mutations can make it more virulent and successful,
I
but
see.
others can also diminish it.
Okay. But
The
why
same
is
was
it
the case
so?
for the COVID.
Yeah, exactly. That is also true, sir. But why is it so that that Mycobacterium leprae, it is so fatal, at the same time it is also evolving drug resistance? In that way, maybe we really have to think about in some other direction where the antibiotic resistance evolution is also happening at the one end. On the other hand, it's also getting virulent. So it's not like gaining virulency is not always dependent on, maybe they are two different traits. I actually still, I'm a little bit also confused, and we actually suffer on this dilemma a little bit at our institute as well, like whether there is any distinct pattern in these two traits or not.
I see.
So, antimicrobial treatment or the drugs actually facilitate selection only.
Sure.
It has been proven that some of the drug resistant mutations were present even before drugs were ever discovered through natural spontaneous mutation also.
Okay.
Only thing is that during the drug treatment, we select the ones which can survive and only those reproduce. So let's say if initially only 1% of the population was resistant to rifampicin, but during the irregular treatment with rifampicin, only that 1% population will have a chance to grow, even if they have a lower division rate for whatever survival cost or the fitness cost. They would replace the sensitive ones. So gradually they would become 90% and the sensitive one will remain only 10%.
True.
But if you are using multi-drugs, then it is very less likely. When you are using multiple drugs, then even if resistance to one drug appears, there might be still other targets to which different target of other drugs which can still kill the bacteria. So that is the principle of having multi-drug therapy in leprosy and tuberculosis and several other drugs, several other diseases.
Like you are targeting multiple targets at the same time.
Yes, so that the chance of emerging multiple mutations would be lesser.
Less likely. True.
The same time.
True, sir.
But if it is one by one, then in tuberculosis now there are strains which are resistant to 14 different classes of drugs because of the stepwise selection. So irregular treatment and different suboptimal regimens are the main reason for such emergence of drug resistance.
I agree. Then sequential treatment may not really matter in that way. That is true. Thank you. Thank you for your insights. Thank you so much.
Thank you. This was actually the question that this was also one of the questions that shaped my career, because I asked this question to Professor Stuart Cole in December 2006, and in just 2 years after, I was investigating the same research question in his laboratory.
Since that, the question hasn't been solved so far. We are still looking for the answer. Honestly, yeah.
Yeah, we have more questions since then.
Yeah, that is for true. The more research we will do, the more question we will get for sure. There is no doubt in that. Yep. Thank you.
Absolutely. Thank you, Surajit. Thank you, Pushpendra sir, for good interaction. So, I'm very sorry to intervene here now, but like we had huge response of people, huge questions. I couldn't accommodate all the questions from everybody, but I tried my best. We still have many more questions, but I think we should have another podcast session later. Definitely, sir. So I would like to thank you, sir, for accepting my invitation on the very first go and getting gel up with this setting of podcast and not the conventional way of talk. So also thank you so much for agreeing to this, my request. I hope you enjoyed it.
I'm very much thankful to you and all your team and participate their time to join.
Sure,
So it's
sir.
difficult on a Sunday to
Yes.
spend time for other activities. But
Yeah.
I think this type of interaction and these type of hobbies are very important really, to get to
Yeah.
know different aspects from we read in research papers.
Yeah.
Thank you for this type of and spending time for such activities there and you and your team.
Thank you so much, sir. Sir, it would be a pleasure that if you could also recommend us few other scientists where we can invite them and have one-on-one podcast with them. So that would be also interesting.
Sure, sure, I will.
I will, I
Thank you
will,
very
I
much.
will definitely write you an email. So wrapping up today's event, I have also sent the feedback form link to everybody. So hopefully we get the feedbacks and we made some mistake, we improve next time. And yes, so thank you so much once again and goodbye to everybody. Thanks for joining on the Sunday
Goodbye
morning.
and all the best to all of you.
Before signing off,
Have a nice day. Bye.
Bye,
excuse
sir.
me
Have a very
everyone.
nice day.
Before signing off, I, Soumya Mishra, on the behalf of Psychom Synapse, would like to request everyone to like, share, and follow on our YouTube channel
Yes,
so that
please.
with the help of you, we can grow and reach to the summit of success. Till then, think,
Yes.
grow, and share.
Bye bye.
Thank you, sir.